Hello, everyone, and welcome to our online lecture series for Biology 2. Today, we embark upon the next major chapter in this course, evolutionary biology, and ultimately a survey of biodiversity. We're going to be utilizing all the information that we've built upon in terms of our understanding of genetics and how genes transmit from individual to individual in different populations. And now taking a long-term view of how those traits that are held in individuals change in populations over time, and combine that with our understanding of novel mutations to help us better piece together how adaptation, natural selection, and in cases where fitness is absent, drift play a role in shaping modern populations. I'll make no secret of it that evolutionary biology is certainly my favorite field of biology. This quote by Theodosius Dobzhansky best sums it up, that nothing in biology makes sense except in the light of evolution. Dobzhansky was a biologist. He had some pretty major cool contributions, and also very eloquent in his advocacy for how a better understanding of evolutionary biology can influence and improve our understanding of the modern world. And this has certainly come true in so many different ways. Now, when we talk about topics like vaccines or medicines or even simple ecosystem design, evolutionary biology shapes so much of it. We improve our understanding of how to manipulate the natural world around us if we understand the evolutionary forces that drove that world into existence in the first place. Evolution gives a reason of why to the world around us. So we'll start our discussion of evolutionary biology by looking at one example, perhaps the best known example of evolutionary biology: Darwin's finches. Now, these finches are from the Galapagos Islands, a chain of islands off the coast of South America. Particularly close to the country of Ecuador. And to set the stage for this situation, we know that there are populations of finches on the mainland of South America, and that these populations of finches on the mainland of South America differ from the species that are found on the Galapagos Islands. Now, the reason why they're called Darwin's finches is because they were first noted by the botanist and naturalist Charles Darwin, and they helped inform his understanding of how evolution can take place over long stretches of time. But before we get there, let's take a look at what his actual observations were. So in looking at the beak size for the finches on the mainland, we will again cartoonize everything, so these are not real numbers, but rather illustrative points. We see that the beak size for the finches on the mainland is 3 inches, 3 centimeters. Units in this case are arbitrary because it's a cartoon, not usually the case in science, right? But we know now that this beak size has a correlation to the underlying genetics. So for example, let's say that beak size in finches is determined by 3 genes: the A gene, the B gene, and the C gene. And those birds that have a beak size of 3 on the mainland have some variability. It's an average of 3, but that does not mean that every single bird has a beak size of 3 centimeters. This underlying genetics can be caused by alleles at each of these loci. Looking back at our quantitative genetics Punnett squares, we can say that the A gene has a variant that adds 1 centimeter and a variant that adds 0 centimeters. The B gene has a variant that adds 1 centimeter or adds 0 centimeters, adds 1 centimeter or adds 0 centimeters at C. And so you get this variation in alleles that add different lengths that give you the range of beak sizes in these finches on the mainland. We see that the original population has an average beak size of 3 centimeters, but that because these genes can segregate in meiosis, or these alleles can segregate in meiosis, and you can get offspring that have much larger and much smaller beak sizes. You can get beaks that range from a size of 0. I should have actually put this on the other side, so let's go ahead and fix that. Sorry about that, y'all. We can get beaks that range in the size of very small, something like 0, or beak sizes that range in the very large, like 6 centimeters, but that you're going to get a distribution around that mean of 3 centimeters. Now, if that's the population that exists on the mainland, and this population were to fly over and visit these different islands, perhaps in different bouts of radiation, you originally would have started with a population that had a beak size average of about 3 on each of these islands: San Cristobal, Floriana, and Pinta. The 3 random different islands I selected from the Galapagos chain. Now, if you look today at these beak sizes, their average is no longer 3. San Cristobal has a very small beak size, approximately 0. Floriana has a huge beak size of 6, and Pinta has a beak size of 4. All different from their original beak size average of 3. How did this happen? Why did these beak sizes change? Well, you can imagine that in this original population that landed there, you might get that on Floriana, that the original population still had that beak size of 3, but because of selective pressures on that island, like large nuts, large things that the birds need to crunch down, the birds with a very small beak would not propagate, and instead the birds with a large beak would have a selective advantage. That selective advantage would cause them to have more offspring. That selective advantage would result in a higher fitness for those birds with large beaks. So you would get a shift in the average beak size to those birds on Floriana to come closer to a beak size of something like 6 centimeters. Now that is true because of the large nuts that are present on Floriana in this example. But we might see something different happening on Pinta. On Pinta, rather than having these extremely large nuts, the food reserves represent what we expect to find on the mainland, where the original beak size of 3 centimeters is our average. But this time, some of these smaller nuts are not present. So you can get a different selective pressure in medium/bigger nuts and fruits, helping skew the new distribution on Pinta to an average of 4 centimeters. And so over the course of a couple generations, and because of this selective advantage in fitness, we might see that from one population, granted, one huge population on the mainlands, because the mainlands will definitely support more birds than each of these tiny islands, you would get this dispersal, this adaptive radiation, where on each island, different beak sizes would do better based on the foodstuffs that are present, therefore leading us to nowadays see these massively different beak sizes that occurred from a once upon a time large homogeneous population. Now, those are the conclusions that Darwin made in observing these different birds on these different islands. He saw their phenotypic variation and interpreted it as descent from one common population that had variability, like we see in our original population, and because of that genetic and phenotypic variability, you can get adaptive radiation. Or the idea that from one population, you can get lots of different beak sizes. They radiate to many different environments, adapt to those environments, and then can eventually form novel species. But that leads us to ask, what exactly is a species? So here's where I digress a bit to discuss the species concept. At what point in that process did Darwin think that these different birds on different islands actually constituted separate species? After all, if they came from one main population and then just spent time separate on different families, as different families on different islands, couldn't they all be considered still part of the same species? How much time or selection or random change would have had to occur in order to push these different bird families to be different species? In order to assess that question, we have defined, and again, humans have defined, meaning that these are imperfect definitions of species, but we've defined species in several different ways. I'll familiarize you with 3 of them. First, the morphological species concept. Second, the biological species concept. And third, the phylogenetic species concept. And we do have to go over these as a group to kind of understand where they're similar and where they're different. In the morphological species concept, you just have species or individuals that look different and are therefore different species. Now, this comes alongside a whole bunch of other caveats, like that these phenotypic differences have to be maintained in large numbers of individuals. You can't just have a single individual being defined as a separate species because they look different. These have to be maintained traits across large segments of the population. But there is no further underlying biology other than appearance, which is a manifestation of the genetics. Remember, it is that genetic variability that leads to phenotypic variability that then can lead to adaptive radiation. If you're not just interested in how things look and perhaps want a little bit more of a concrete answer, something more aligned with traditional biology, we have the biological species concept. And yes, this is kind of the original definition, hence the grandiose name of the biological species concept, but it's fairly intuitive. In the biological species concept, different species are defined by the inability to make fertile offspring. So, this works very well for currently living species. We can obviously take different groups of animals and see that in the wild they will never produce children, and therefore that these species are truly different species. For example, if you have a cat and a dog in the home, one is a male, one is a female, they will actually never mate, contrary to the cute viewpoints of some small children. I have definitely heard some small kids, myself included, assume that dogs were always male and cats were always female and they always fought. But this is certainly not the case. These different species never produce hybrids that are viable, never even attempt to produce hybrids, and therefore are defined as different species. In the same way, if you were to, in a laboratory, take human sperm and a chimpanzee egg, you would not be able to produce a fertile offspring. It is therefore, by the biological species concept, that humans and chimpanzees are separate species. In the phylogenetic species concept, which is probably the one that we'll be revisiting a bit more later when you understand a bit more about phylogenetic trees, we look at how these groups cluster. And it is clustering on branches that allows us to determine whether these species are truly separate or not. For example, if you take a genetic alignment of the entire genome of two different families of chimpanzees, and they cluster in such a way that chimps from family A are all over here, and chimps from family B are over here, all clustering together separately. This would suggest that these are two separate species, at the very least, two separate families in this case. But if instead you have a mix of A's and B's clustering together on each other's branches based on those genetic alignments, you would instead consider this whole group to be a single species because of genetic mixing. And that's ultimately what the species concept relies on. Some sort of prevention of genetic mixing. Because separate species will no longer be able to exchange their genetics. So how do these different species come about? How do we go from one species on the mainland to several different species in the islands that are now biologically distinct, morphologically distinct, and phylogenetically distinct from the species on the mainland? So here we'll go into a couple of different vocabulary terms. The first two have to do with region of speciation, that being allopatric and sympatric speciation. In allopatric, we have the two roots, allo or other, same as in allele, different versions, and patri, meaning fatherland. Species that have speciated allopatrically have done so in different places. Physically separate from one another, that physical separation has actually caused a lack of gene flow, a lack of genetic mixing. Since I'll be using this a lot, gene flow is the true biological way of saying genetic mixing between populations. That separation in physical space has caused allopatric speciation. And in the case of birds, where these islands seem to separate these birds based on the winds, it's quite easy to follow the wind and fly out to the islands, but if a bird wants to fly back, that's a no-no because the winds will frequently not allow them to do so. In other cartoon examples of this, you might expect one homogeneous population that at some point down the road may be split in half due to a new river forming in the area, and then all of a sudden you've got the individuals that live here being genetically isolated from the individuals that live on the other side of the river without that genetic mixing, simply due to enough time, as we'll talk about later. Both time or adaptation can drive evolution. You would get allopatric speciation in these two populations that used to be one. In sympatric speciation, you keep the species in the same fatherland, meaning that somehow, in the same place, we got speciation to occur. So if it isn't different physical boundaries, like in allopatric speciation, sympatric speciation relies much more on potential reproductive barriers. Meaning things that might keep different individuals of the same species from mating with one another. The first major reproductive barrier can be something as simple as assortative mating. Assortative mating is something that certainly occurs within these birds, the Darwin's finches, but also occurs in many other species. Assortative or assortative mating is the idea that mate preference can lead to genetic isolation. For example, let's say that there is some genetic region that causes a human being to be hardwired to only want to mate with individuals with pink hair, bright pink hair. In this case where pink hair is such a strong trigger, they might prevent mating with any other individual and keep mating with this particular phenotype. So even in the case where all these individuals are found in the same place, the gene flow is not maintained between everyone else and the small subset of the population that mates only with those individuals with pink hair. This selective pressure to mate with only those with pink hair can eventually cause this subset of the same population to speciate. Now that change has to happen over long periods of time, and it is often through biological processes of reinforcement, which is any kind of change that helps keep two populations separate in order to enable them to speciate. And these reproductive barriers that we're going to talk about in a second are great examples of different types of reinforcement. They generally come in two different flavors: the prezygotic, meaning stuff before baby making, before the baby is conceived, and then the postzygotic, meaning that things that occur after conception that prevent this individual from developing into a fertile offspring. These prezygotic barriers can be things like habitat. In one example, you can see frogs that live in tree trunks versus in the marsh. And in this cartoon example, the fact that these different groups of frogs that are all of the same species have really specialized on different environmental niches, different ecological niches, even if they live in the same area, maybe can even see each other, this prezygotic barrier of habitat differentiation can prevent them from actually ever even attempting to conceive a baby, even if they biologically could. In temporal mating, or temporal barriers, the timing of mating does not match. This occurs within species of palms. On a particular island in the Pacific, the Lord Howe Island. In this one species of palm that arrived on the island something like 4 to 6 million years ago, we saw that this group of organisms flowered all at the same average time. But over disparate time frames, 4 to 6 million years, very far apart times, we started seeing that some of these palms would flower earlier and some of these palms would flower later. Now, the palms that flower early kept pushing for more and more earliness, and the palms that flowered later kept flowering a bit later in the season. And in this way, you got isolation. If you're expecting to mate in the summer, and another member of your same species is expecting to mate at the middle of winter, there's no opportunity for gene flow to occur between these two different populations. And in this way, another prezygotic reproductive barrier will form. Next up, we can have behavioral. These are well illustrated in organisms like the blue-footed booby or in cichlids, or even in Darwin's finches. You can get things like swim patterns in fish, meaning do you swim deep below the water when you're anticipating a mate? Do you swim at the surface when you're anticipating a mate? Or in some of our bird species, you can get mating calls that are specific. If you are waiting for a very specific rhythm in order to wait to find your mate, and this different population has changed its rhythm, you will probably never mate with them because they're not making the right mating call. It's not the right courtship. Another barrier, starting to get a little bit more on the physical incompatibility side of things, we have a mechanical barrier. This is where the reproductive components are inaccessible. For example, in different populations of damselflies, these insects, you've got the length of the reproductive structures as separating different populations of damselflies. If reproductive structures cannot reach where they need to reach, you can cause reproductive isolation all before conception occurs. And our very last type of prezygotic reproductive barrier is that the gametes don't recognize each other. The sperm and the egg do not fuse. So even if, all things aside, you're mating at the right time, you're making the right call, you are mechanically able to do so, and you live in the same habitat, if you take sperm and egg from these two different populations, and the sperm never fuses with the egg, you now have a prezygotic gamete barrier. And this is very well illustrated, and is oftentimes a point of reinforcement in a lot of different species. For example, taking human sperm and chimpanzee eggs, never going to fuse. Can't force it to happen. Therefore, we have a barrier that reinforces our separation as a species. Our postzygotic barriers are after conception. There are two different flavors of this. The first is hybrid inviability. Like where the offspring is actually made, the sperm and egg fuse, the zygote formed. But in development, that zygote miscarries or aborts because there is some developmental change that is not compatible between these two different populations, therefore causing them to never create viable offspring. Now, I want to highlight that this may have occurred with some of our different hominid species, as we discussed recently. We may have seen mating be possible and zygote formation be possible, but due to some incompatibilities, potentially even mitochondrial, some of those directions of those crosses may have resulted in inviable offspring. On the flip side of things, you might even get an individual that is viable. You get hybrid viability, but sterile hybrids are still a postzygotic example of a reproductive barrier. The best known hybrid sterility example occurs when you take a horse and mate it with a donkey. You can force it to ignore the habitat differentiation, the temporal variation, the behavioral variation, the mechanical variation, and even, and there is not a gamete inviability between these two, and the organism will form, the progeny will form to make a mule. But the reproductive organs of a mule are not formed. Therefore, a mule cannot mate with another mule ever. And because this line ends with the mule, even though the mule is a biological organism, it is not considered a separate species. It is a one-off in terms of mating pattern from a horse and a donkey that then cannot continue its line. This postzygotic barrier would cause mating events from these disparate populations, these two different populations, to not be favored. If you can't make babies, you're probably not going to be able to continue on your baby lineage, right? So all of this goes to say that there are a lot of different forces that can drive these evolutionary patterns that we're seeing in things like Darwin's finches. We know that the underlying genetics has a big role in local adaptation, that local environments can shift the different selective pressures to cause shifts in phenotypes of our different species. And now we have an underlying background for what a species actually is and how some of those species can actually form. So let's now go ahead and take a look at some of the vocabulary tied directly to the theory of evolution. Evolution can be defined very simply as allele frequency change over time. And this is one of the major misconceptions that we're going to address. Evolution has no purpose, or at least higher purpose. Evolution has no point other than survival. And evolution has no goal. Evolutionary changes can occur because they are adaptive, and when they do so, we call them evolution by natural selection, or they can occur by random chance. When that occurs, we call it genetic drift. In order to understand how evolution can occur by random chance, I'll introduce you to another short vocabulary term called a population bottleneck. And to illustrate this type of evolutionary change, we'll go back real quick and revisit our finches. Say hypothetically that of the finch migrants, the first individuals that made this travel from the shores of Ecuador to San Cristobal, let's say that that group of finches had only very, very small beaks. The immigrants, by chance, were from the lower distribution of beak size from the mainland. That means in a single generation, you would only get small beak sizes. No adaptation required. This bottleneck you've essentially set out to ignore, you've essentially set out to ignore the rest of the distribution and focus only on the small beak sizes just by random chance that that happens to be the group that's immigrating. Now again, chances are that if you're going to sample a random set of individuals from that species to travel, you're going to get a wide distribution. But if you're only picking 5 immigrants, dumb luck or genetic drift can cause this change in allele frequencies in as short a time as a single generation. So in that way, evolution can be driven either by adaptive or stochastic forces. Or random forces, truly random forces. Now these observations about evolution were first put to pen and paper by Charles Darwin and his voyage on Her Majesty's ship, the Beagle. Darwin's voyage on the Beagle set out in 1831, late 1831, just after Christmas, and lasted 5 years to 1836. Quite a long time, but in that journey he went from Plymouth in England towards the Azores in the Atlantic Ocean to Cape Verde, down to South America, visiting a lot of different places, including Rio de Janeiro and the Falkland Islands, and then locations on the western coast of South America, including Valparaiso and the Galapagos. He then continued on to New Zealand and Australia, visiting a couple places in the Indian Ocean or along the Indian Ocean as well, before stopping in Cape Town, South Africa, and then returning to Plymouth, all over the course of 5 years. Now, in these journeys, he made a large number of observations. His major observations would later be known in the field of biogeography, or our understanding of how current location has been shaped by past location. In simpler words, the idea that Darwin found similar animals on different islands or continents. He observed that in a lot of these places that he visited, a lot of the plant life was the same, but they had small differences. Or a lot of the animal life was the same, but that they had small differences as well. He used these observations on animals like the finches or tortoises or plants to put together the idea of divergent evolution. The basic concept that from one species you could get natural selection that would drive this one species and its many subpopulations to eventually form many distinct species. And this is the mechanism that we discussed in the finches, but also the mechanisms that he saw in tortoises, different llama relatives, and in particular in some of the larger mammals that he saw, Darwin also tied together the fossil record with his concepts of biogeography, putting pen to the idea that you could actually get evolutionary changes from a species that exists only in the fossil record to a species that exists in the modern day that descends from that single species in the fossil record. Meaning that you could get descent with modification. He also noted that there were examples where very different species ended up looking very similarly because they occupied similar ecological niches. He contrasted this idea of divergent evolution with convergent evolution. The idea that from many distinct species, you could get a common form. Meaning, even though mammals and birds are very different, mammals that have evolved for flight, like bats, will have wings similar to birds that fly. But the structure of those wings will be quite drastically different. And this becomes very apparent when you start looking at the morphological evidence, as Darwin was doing on his 5-year voyage. He had a tendency to want to eat many of the different animals that he collected. He wanted to taste test them. And so in doing, he would find out the underlying bone structure. Because if you're cooking a lot of these things, a lot of the different animals, you would oftentimes take apart the bones. And he started noting that the bone structures between animals like humans, dogs, birds, and whales all have the same count, even if the structure and the length and the location of those bones is fairly different. A bird's wing is made of its entire arm, and you still have the 2 bones of the forearm with the metatarsals at the front. Now you get those wrist bones and the 2 bones of the forearms in humans, but they look very different, and even those wrist bones have a quite different structure. Dogs are more similar to humans, and whales are quite distinct. So you had this morphological evidence of descent with modification and homology, or shared ancestry. All of these organisms, humans, dogs, birds, and whales, all inherited their ability to make a forelimb, an arm, from the same common ancestor. They each adapted to different areas, different ecological niches, and were therefore selected to do very different things. Homologous traits that provide shared ancestry are examples of divergent evolution. Analogous traits are instead traits drawn by shared function, like the bat versus the bird wing. Now, I am tying in some of Darwin's conclusions with more practical modern-day examples. So note that if you go back and read his exact text, which I strongly encourage you to do, they are fascinating and were quite instrumental in my own thought formulation as a biologist. But we also need to keep track of a lot of different vocabulary terms and 100+ years of synthesis of Darwin's ideas. So we got homologous and analogous traits analogous being those that correspond to convergent evolution. We also saw Darwin tying in some fossils, but he also tied in the fact that over geological time, that Earth changes quite drastically, suggesting that there might have been massive change into different environments. His evidence for this change over geological time was that he found seashells buried naturally at the top of the Andes Mountains in South America. Now, how on earth do seashells that are typical of the bottom of the ocean end up in the Andes on the top of these really, really tall mountains? Well, Darwin concluded that those seashells got there because many millions of years ago, the Andes were actually underneath the ocean floor, and that plate tectonics, or shifting continents, would have potentially pushed these mountain ranges, or once upon a time seafloors, up and up and up to become mountain ranges. So in this way, he was tying together biogeography, fossils, and some of his morphological evidence. His last bit of morphological evidence comes from traits that are called vestigial traits. Vestigial traits are body appendages that are no longer used as first intended. Now, one of the best examples of vestigial traits in humans is your tailbone. You as a human no longer have a tail, but you share ancestry with a wide variety of different monkeys that do have a tail. A prehensile tail that's able to grab things. You still have that bone from which the tail would attach, but no tail grows. Hence, the tailbone is still a vestigial structure. Another, less thought about vestigial structure are some of the tip points in our ears. Now, humans generally do not have the ability to move their ears around a lot. Contrast that to some of our monkey relatives, whose ears will fly around in the direction of sound in order to better catch it. Just go ahead and say the word dinner around my dog, and you will see how much muscular control there can be in controlling that ear. Now, we still have that tip of the ear to which muscles would have attached or protruded from, but we no longer have the ability to move the ear, as we see in some of our monkey relatives. So with his ideas of morphological evidence and fossil evidence, Darwin was able to put together his theory of evolution that organisms change over time based on both adaptive evolution, natural selection, and random genetic changes, genetic drift. Now, just for funny story time about all this, Darwin's theories, even though he finished the voyage of the Beagle in 1836, he spent quite a long time sitting and stewing on his ideas. In the most epic case of procrastination, Darwin wanted to work on this till his theories were completely perfect. However, he wasn't the only one thinking about this. In 1858, he received a letter from a colleague named, so this is Darwin. I want to make that clear that I'm not labeling this as someone else. This is indeed Charles Darwin as a younger man. But he worked alongside another individual named Henry Wallace Alfred Russel Wallace, not Henry Wallace, who sent him a letter that basically described his ideas of descent with modification and natural selection in 1858. Well, after procrastinating on his publication for something like 20 years, So definitely reevaluate your procrastination tendencies if it's taking you 20 years to put together your thoughts. They decided to publish their work together. Now, Darwin gets a lot of the credit for this because he was quote-unquote first to this, but I don't want to diminish the fact that Wallace's discovery was independent with a lot of the same observations, a lot of similar threads in his data. So, with work published, Darwin went on to continue to influence evolutionary thought by publishing further works on the descent of man, the expression of emotions in man and animals, and then he worked a lot with different plants. And so Darwin's work really solidified our understanding of evolutionary biology. It really put together this image that from a single founder species, we could get adaptive radiation over long periods of time to create a much wider, diverse array of current life. And so nowadays, we have gathered tons of incredibly strong evidence to put together our theory of evolution. And I don't use theory here in the way that most folks say, or rather non-scientists say. The theory of evolution is not just a theory. It is our best biological hypothesis to put forward that we have no contradictory evidence, no truly opposed evidence for. It is our best idea without being refuted to date. So a theory is actually a very strong statement. It is not a fact in terms of theory of knowledge, but all of our evidence supports that evolution is real. And what is our evidence? Well, as a quick recap, we have that biogeographical evidence from Darwin and many other observations that we see slightly different species on different islands and continents corresponding to their relationships. And we even can draw this out to older time periods like Pangea, when all the continents on planet Earth were smushed together. We have beautiful fossil evidence that gives us good corresponding morphological evidence of both homologous traits and, in some cases, analogous traits. We have excellent biochemical evidence nowadays, where we look at protein conservation, meaning that a lot of the proteins that are present in our bacteria still share some similarity to proteins that are found in us. And when you look at, for example, vertebrate lineage species, you only see that of all the different protein families present in animals that have a spine, only 7% of them are brand new. So a vast majority are just innovations on what used to be there. We also, in addition to our massive biochemical surveys of protein conservation, have DNA similarities, which will be a huge focus of the different labs in this section of the class. Now, I would be remiss in my discussion of evolution without discussing some of the historical criticisms. So we'll take this as our last major point in this lecture. Some of the criticisms of evolution are that we lack transitional fossils. The idea that we should have a missing link for every different branch point, every different morphological change for example, from the common ancestor of something like chimpanzees and humans. But the problem with that is that transitional fossils always show a direct progression. And the odds of finding some of those transitional fossils are incredibly low. So many different species would not have left fossils if they did not die in the right place, in the right preservation. We do have some transitional fossils, such as some of the lineages of dinosaurs that had feathers that then would have evolved into the lineage that we know as birds today. But the fact that we do not have every single transitional fossil does not mean that evolution is false. A lack of evidence is not damning evidence. Second, stems from the fact that evolutionary biologists state that evolution is oftentimes random, and that because evolution is random, it could not produce life that is so complex. Well, again, this challenges the human imagination. I understand that it might be difficult to think about the evolution of a complex structure like the human eyeball, but understand that the evolution of things like the eye occur in very small steps. You get progressively more and more complex eyes that can detect more and different colors of light from an organ that was not able to really detect much light at all. Because evolution occurs in these small steps, you can get massively complex things like the eyeball. For example, in modern human society, looking at an automobile like a Tesla, you would not conclude that this Tesla was created from whole cloth, from nothing, but rather you could look back in time and see how an electric car like the Tesla used the blueprint of a gasoline-driven car that then used the blueprint of a gasoline-driven car from an era before, from an era before, from an era before, that then draw their structure actually from horse-drawn carriages, that then draw their structures from wagons. So you can draw this connection from a Tesla to a wagon being pulled back in 3000 BC. The line is kind of direct. And in the same way, evolution makes these small steps. You don't just go from a horse-drawn wagon to a Tesla. You have to go through all of the small iterations in between. The third criticism is that evolution is not testable. And to that, I say it totally is. We have really cool experiments in different research groups across the world where we've tried to evolve new digestive systems for bacteria and have been successful in doing so. We've looked back at how different evolutionary pressures might have caused different evolutionary changes and tried to recreate or simulate some of these. Now, of course, in small organisms like bacteria, we're able to do this much faster, but just because evolution occurs in bacteria does not mean that evolution does not occur in some of our larger mammals. We just don't have the time to be able to recreate a lot of those experiments. So evolution is absolutely testable as a hypothesis, and we've seen it done over and over. Our fourth criticism is that it is just a theory, and to that I say, nah, it's a pretty solid hypothesis. It's a pretty solid statement. It's as close to fact as we can get. Our fourth criticism is that what about the origins of life? How do you begin having life on planet Earth? Well, because we don't have a time machine, we don't exactly know, but we can see the patterns of change that are occurring in evolution, and we can even start to see that there are different patterns to simple unicellular life that we can kind of recreate in different laboratory environments. Again, future scientific discovery might shed better light on the origins of life. For those of you who are interested, I'd recommend the Miller-Urey experiments that show that from nothing, with the addition of a little bit of electricity and some clay, you can get some of those early macromolecules that life relies on. So it is possible that science will continue to better understand the origins of life on planet Earth, and that it is indeed possible to go from elements to self-replicating elements like bacteria. The fifth criticism is that evolution shows that there is no, or rather that evolution is trying to achieve a purpose. And to that, I would say that it is not. There is no purpose to evolution but current survival. We oftentimes see lineages go extinct because they may be adapted very strongly to the current conditions, but then when those current conditions change, they're no longer well suited. And so evolution is not trying to create a perfect being, but instead create an individual, a population, that is well suited to produce the most offspring in the current conditions. Because those conditions change, we get a lot of extinction. And there has been a huge amount of extinction in the history of all species on planet Earth. So, hopefully that kind of puts together a lot of the different vocabulary and ideas that you've heard behind evolutionary biology. I want to encourage you to devote this statement about evolution being allele frequency change over time to memory, because we will be letting go of some of our allelic focus, as we've done in the last section on genetics, and instead seeing how these alleles change over massive time frames to result in different species. In particular, next time, we're going to look at how concepts of gradual speciation, whereby you get stepwise changes that result in different species, or punctuated equilibrium, whereby the branch just occurs fairly immediately from that single founder species, but ultimately both of these allow you to draw connections between modern day species through time in the past to some sort of most recent common ancestor. Something that was potentially a founder species once upon a time, but no longer exists today as it did in the past. And that's absolutely critical to understand. Humans did not descend from chimps. Just like the amakihi and the Nihoa finch did not descend from, or the amakihi did not descend from the Nihoa finch. They are both independent branches from a shared most recent common ancestor. This principle, or this observation of phylogenetics, will be the focus of our next lecture. So I will see you then.